The regulation of transcription in eukaryote is in general much more complex and less well understood than the prokaryotic mechanism. There are three types of RNA polymerase in eukaryote. One for rRNA working in nucleolus, two for mRNA, and three for tRNA and 5S rRNA. The latter two working in nucleolus. We're gonna here focus only on the type 2. There are different combinations of specific DNA sequences, each of which is recognized by a corresponding site-specific DNA binding protein called transcription factors. Each combination of DNA sequence and cognate transcription factor constitutes a control module. The DNA part of each control module can be divided into three main regions: core basal promoter element called Tata box, position 25 base pair upstream of start site. Second one, promoter proximal element, sometimes called response element, position near to transcription initiation site. Third, enhancer element that could be positioned even 20 kilobase pair apart. Transcription factors can be divided into two classes: general factors of pre-initiation complex are required for the expression of all structural genes transcribed by RNA polymerase II, so that it could be ubiquitous. Second one is a specific transcription factor that binds either to promoter proximal DNA sequences or to distal enhancer elements. Pre-initiation complex composed of TF2D, 2A, and 2B are essential for the initiation of transcription. Pre-initiation complex binds to Tata box, recruit RNA polymerase II, and also recruit specific transcription factors. 2F2D is key link between specific transcription factors and general pre-initiation complex. There are TVP protein in this complex, which is Tata box binding protein plus TVP associated factors, TAF2. There are many various kinds of factors in this category, but due to the instability and heterogeneity of this complex, details are not determined yet. Tata box binding protein is critically important in initiation. We are looking at the consensus sequence of Tata box, Tata and Tata, 8 nucleotide long. This is from the East, and this is from the Arabidopsis species. yeast TBP is 27 kilodalton, has two domains. C-terminal actually composed of two domains and highly conserved. C-terminal itself could be functional for DNA binding and transcription. Its structure was determined in 1993 by Paul Ziegler in Yale University. You're looking at side view and bottom view of this structure. DNAs are positioned here. So similar to set forms with the strap, this strap. Topology says each domain 4 beta plus 1 beta for the DNA binding, this region here, the bottom view. And in addition, you can see each domain has 2 alpha helix and loop connecting two motifs, which could provide site for the interaction with TF2D and other specific transcription factors. Okay, you are looking at DNA TBP complex. See, there are some distortion in DNA structure. DNA actually was bent by 100 degree by two DNA kinks at 1 and 2 and 7 and 8. And this region corresponds to the minor groove, but this region was widened as a result of binding. And also there are some unwinding of DNA was found in 3, 4, 5, 6 base pair. DNA binding to minor groove also prominent, which is a contrast to other the binding proteins, where you could see mostly by alpha helix or a pair of antiparallel beta to major group of DNA. So in details, in general, large hydrophobic interactions coming from 15 side chains of from 8 beta to sugar and base of DNA. In addition, there is hydrogen bonds between arginine and lysine at the edge of protein to phosphate DNA. There are sequence-specific interactions. We could see later the specific hydrogen bonds in minor groove. We're going to look at some each individual features. This 8 Tata box was bent. You see, sharp turn. This Tata box on top, behind the Tata box you can see arrow representing beta protein sheet of Tata box binding protein. In the middle you see hydrogen bond with a red line between protein and base in Tata box. Interesting feature in Tata box is that it has TA pair compared to the GC. TA is less stable than GC, so that it's much easier to separate. Next type will be Antennapedia homeodomain. First time determined by Vitrucci and Carl Pavo. In general, homeobox is very highly conserved for their size, 20 amino acid, function as DNA transcription factor. Actual structures are 3 helices, second one and third, similar to helix-turn helix. Almost identical to prokaryotic helix-turn helix, second and third helix, but the third helix, recognition helix, is much longer than the counterpart of prokaryotes. The binding is different from the prokaryote. Single monomer could bind to major group. The way it binds to is similar to prokaryotic helix-turn helix. Recognition helix binds to the DNA major group. This is side view, showing some specific interactions between two components. Specifically, arginine 53 and asparagine 51, plus some other residues positioned in DNA major group, and also some hydrophobic residues 48 and 49 also plays some important role in positioning between recognition helix and N-terminal arm. N-terminal arm, this one, green one, also binds to the minor group, this region. Arginine 3 and 5 make a non-specific interaction with phosphate. Homeodomain from 4 different types are aligned to show some common consensus. You can see the position of 3 helices, 1, 2, 3, and then the green one forms hydrophobic core, blue, non-specific interactions with DNA, red one, contact with edges of DNA base. So, some regions are highly conserved, especially hydrophobic core. There are so many different types of homeodomains, but the DNA binding sequence and homeodomains are almost identical. So people could ask how each homeobox domain binds to specific DNA.